I'm Professor Sims, and this video is an introduction to my General Genetics lecture course. If you're a student currently enrolled in this course, please consult the syllabus and the course Moodle site for assignments, quizzes, due dates, and other information. This course is broken up into 10 lessons, each of which has specific learning objectives. The learning objectives for this unit are going to be becoming familiar with course expectations, resources, information, and then we'll go into an overview of genetics. We'll talk about the nature of genetics, genetic variation, the central dogma of biology in general and genetics specifically. We'll talk about gene expression and the relationship between genes and traits. And again, it's just an overview of genetics and the research methods that people use, applications of genetics, and even some ethical and social implications. The purpose of this course is to provide students with an understanding of the concepts of classical and molecular genetics. Students are going to learn the Mendelian laws of inheritance, so stuff like genetic crosses, and then also non-Mendelian laws of inheritance. The course is designed to help students apply their knowledge of genetics to solving complex problems. You'll get basic knowledge on the process of the central dogma, mutations, chromosomal aberrations, and things having to do with genetic diseases. The overall course learning objectives relate the structure and function of the DNA molecule to its role in encoding genetic material, understand how to identify and classify mutations in DNA, discuss the repair systems of DNA, principles of inheritance as formulated by Mendel, and basic principles of neo-Mendelian genetics. The textbook for this course is a free OpenStax textbook that you can get online. I also have the PDF posted in Moodle. Here very quickly, I just wanted to show you that the table of contents for this book, it's all hyperlinks. So you don't have to go and scroll all the way to chapter 15, for example. You can just click on the hyperlink that is in the table of contents, and it will jump you right to wherever you need to go. Be sure to spend some time familiarizing yourself with the course Moodle site. We will be using this site extensively, and this is where you're going to find a plethora of resources, including the news forum, the course syllabus, the calendar, your grading rubrics for your written assignments, writing resources like APA guidelines, things like that, and also the slides for all of the video presentations, all of your quizzes, exams, and written assignments. All of that stuff is going to be communicated to students via Moodle. The lesson videos are embedded in the Moodle shells for each lesson, but they can also be found on my YouTube channel at www.youtube.com/c/Professor Sims. Please do remember that these presentations are designed to supplement and not substitute for your reading assignments. The Moodle modules for each lesson is also going to include a quiz. This lesson includes a quiz, and then all 10, like 1 through 10 of the regular lessons also include a quiz. So that's 11 quizzes altogether. Make sure that you see the quiz links and the course calendar for the due dates. So those are always going to be due on a Friday before 5 p.m. Moodle's going to have all the information that you need regarding the quizzes and assignments and all of my policies for grading, late assignments, all that good stuff. So please do make sure you spend some time reading the syllabus and getting familiar with everything on the Moodle site. I encourage students to study smarter, not harder, by which I mean you want to get more out of your study time and you want to learn more in less time. So what I see a lot with my students over the years, I've seen them using these techniques here on the right for studying, things like rereading, rewriting, going over old quizzes, going over old class notes. And that's fine, except it's very, very time consuming, and at a certain point your brain kind of burns out, especially if you're doing so much rereading and rewriting and rewatching that you're losing out on sleep time, because your brain needs sleep in order to form new neural connections and memories. So I would like to encourage students to kind of shift away from these methods here on the right and shift towards these ones on the left, which have been scientifically proven to not only take less time to do, but they're more effective as far as long-term memories and comprehension of material is concerned. So you are going to do some rereading and rewriting, but what you really want to do is fill in the gaps of your knowledge. You need to kind of take inventory of what you do and don't understand and spend your time focusing on the things that you need more help with. So you're going to write things in your own words. You want to test yourself, like write little mini quizzes and tests for yourself and answer them. And you want to try to interleave things. So you want to try to relate the new things that you're learning to things that you've already learned. And you also want to vary your studying in such a way that you're not spending huge blocks of time doing one activity or studying one concept. So you don't want to spend, 4 hours at a time just reading the textbook because, about 20 minutes into that, your brain kind of shuts off. You want to take breaks. You want to alternate between reading activities and writing activities and maybe other types of activities like watching a video or a simulation, or maybe if you're good at drawing and you're a visual learner, maybe you want to make a diagram or a figure or something like that. Anyway, you want to switch it up and make sure that you're studying smarter. So let's get into just an overview here of the study of genetics and what kind of things we're going to dive deeper into throughout the semester. Genetics is the scientific discipline that explores and investigates the heredity and variation of living organisms. It's a branch of biology that focuses on the study of genes, their structures, functions, and how they're transmitted from one generation to the next. Genetic research has revolutionized our understanding of life and provided invaluable insights into various fields such as medicine, agriculture, and evolutionary biology. Genes are segments of DNA. DNA stands for deoxyribonucleic acid, and this is the molecule that's responsible for storing and transmitting genetic information. Genes contain the instructions for building and maintaining an organism, influencing its traits and characteristics. Each gene encodes a specific protein or RNA molecule that performs a particular function within the cell. Geneticists study the structure and function of genes to unravel the mechanisms underlying inheritance and the development of genetic traits. It is important for you to have a clear understanding of the difference between DNA and a gene. DNA is a molecule. It's that double helix molecule that we all know that carries all the genetic instructions. Well, the genes are a sequence of DNA. So genes are within the DNA. They aren't the same as the DNA. There's a lot of DNA that codes for different genes, different traits, and then there's a lot of DNA that doesn't code for anything. So if DNA is the molecule that has all of the genetic information, well, a gene is a sequence within that molecule that codes for a specific protein or RNA that has a specific function. And then once you get the difference between DNA and genes, it's also important to understand the difference between a gene and an allele. An allele is essentially a different version of a gene, which varies according to the nucleotide base present at the specific location on the gene. So it's a variant. It's a form of a gene. Whereas the genes are the segments of DNA that contain the instructions for building the building blocks of a living organism, right? Well, the alleles are the variants of that gene that live in the same location or the same locus on a chromosome. The next bit of terminology that's really important to have under your belt before you start studying genetics is going to be the difference between a genotype and a phenotype. A genotype is the specific combination of alleles that an individual possesses for a particular gene or a set of genes. So a genotype is the genetic makeup of an organism. On the other hand, a phenotype is the observable physical, biochemical, or behavioral characteristics of an organism that result from the interaction of a genotype with its environment. The genotype is the genetic blueprints. The phenotype is how that blueprint is expressed. It's a combination of what's in your genes plus the environment that you grew up in. So your physical appearance, those kinds of things are phenotypes. So in this example here, it's giving the alleles, right? The genes. So this one is a homozygous dominant. Don't worry, we're going to go much more into depth into all of this kind of more specific terminology. But for now, just know that capital B it's a locus that has two of the same allele, and those are dominant for whatever trait you're looking at. Then we have one here that is heterozygous. It has one that is dominant and one that is recessive. And then this one here has two that are recessive. And that is translating into a phenotype where capital B is purple, capital B is purple, and then little b is white. So the genotype are the actual alleles. The phenotype is those alleles being expressed as an observable trait. Gene expression is the process by which the information stored within a gene is used to produce a functional product, such as a protein or an RNA molecule. Gene expression is tightly regulated, and it's a complex process that occurs in all living things. It involves a series of steps, starting with the transcription of DNA into RNA by RNA polymerase. The resulting RNA molecule, known as messenger RNA or mRNA, is then modified and transported out of the nucleus to the cytoplasm. And then in the cytoplasm, the mRNA serves as a template for protein synthesis during translation, where ribosomes read the mRNA sequence and assemble amino acids into a polypeptide chain. Gene expression is highly dynamic, and it can be influenced by lots of things, including environmental cues, cell signaling pathways, regulatory molecules, right? So the precise control of gene expression is crucial for maintaining cellular function and the development and growth and specialization of different cell types in an organism. And this is closely related to the central dogma of biology or genetics. So the central dogma is the theory that states that genetic information, it flows only in one direction, and this is the pathway. So it starts with the first step is DNA replication. The second part is transcription of DNA into RNA. And the third step is translation from RNA into a protein. So this is the order. It's only 3 steps, and you should make sure you know these 3 steps. So the key factor of the study of genetics is inheritance. Inheritance is the process by which genetic information is passed from the parent to the offspring. It involves the transmission of traits through passing of genes. The genes are inherited in pairs, with one copy inherited from each parent, and then the specific combination of alleles that you get from your parents determine your genotype. Genetic inheritance can follow different patterns, including dominant and recessive inheritance. So let's talk about dominant and recessive. Dominant traits, these are the ones where one allele can mask the expression of another. In other words, the dominant trait is expressed even if the individual carries one of the alleles for the recessive trait, right? It kind of overpowers it. So earlier when we were looking at these flowers, right? We've got 2 dominant alleles here with the capital B's, 1 dominant, 1 recessive, and then 2 recessive. So notice that even when you have one of the recessive alleles, if the dominant allele is there, it's going to mask the expression of the recessive allele. Now, recessive alleles can get expressed too, but in order for that to happen, you have to have the recessive allele from each parent. You have to have 2 of them in order for the recessive trait to be expressed. Now, I can't believe I've gotten this far into an overview of genetics without mentioning Gregor Mendel and his work. I think this is a good place to show you guys this short video that gives an overview on Mendel and his works as they pertain to genetics and genetic inheritance.
The science of genetics began in the 1800s when Gregor Mendel, father of genetics, figured out how traits are inherited by studying peas. Gregor Mendel discovered the fundamental laws of inheritance. He said that genes come in pairs and are inherited as distinct units, one from each parent. He recognized the mathematical patterns of inheritance from one generation to the next. Mendel's laws of heredity are usually stated as 3 points. The law of dominance. The first law states that an organism with alternate forms of a gene will express the form that is dominant, meaning a dominant gene will express itself over the recessive gene. The law of segregation. It states that each individual that is a diploid has a pair of alleles, a copy for a particular trait. Each parent passes an allele at random to their offspring, resulting in a diploid organism. The allele that encloses the dominant trait determine an individual's observable trait, such as dominant hand, eye color, and blood type. The law of independent assortment. It states that the alleles of 2 or more different genes get sorted into gametes independently of one another. In other words, the allele a gamete receives from one gene does not influence the allele received from another gene. Mendel's laws allow you to understand how character is inherited and what determines the phenotype that different individuals acquire.
Between 1856 and 1863, Mendel conducted hybridization experiments on garden peas, and he chose some distinct characteristics of the peas and then conducted cross-pollination or artificial pollination on the pea lines that showed stable trait inheritance and underwent continuous self-pollination. Such pea lines are called true breeding pea lines. He selected a pea plant for his experiments because the pea plants can be easily grown and maintained. They're naturally self-pollinating, but can also be cross-pollinated. They're an annual plant, so many generations can be studied within a short period of time. The pea plant has contrasting characters that can be observed. So if you look at things like seed, color or form, flower, pod, stem, they have these different characteristics that you can look at and see how they are passed down and how they are not. And what Mendel found was that certain factors were always being transferred down to the offspring in a very stable way. And now we know that those factors that he saw are genes, and that the genes are the units that are being inherited. Genetic variation refers to the differences that exist within and between populations. Genetic variation is often caused by mutation, and there's other factors that we'll talk about later in the semester, but the variation is the driving force behind evolution because it enables organisms to adapt and survive in changing environments. Geneticists study variations in genes like mutations or polymorphisms, things like that, to uncover the effects on individuals and on populations. The understanding of genetic variation provides crucial insights into things like disease susceptibility, drug responses, and overall population genetics and dynamics. Genetic research employs a wide range of methods and techniques to study genes and their functions. And these include things like DNA sequencing, which determines the order of nucleotides in a DNA molecule, and that allows for identification of genes and variations within them. Another example is polymerase chain reaction, or PCR. PCR is used to artificially amplify DNA in a lab, so you can target specific DNA sequences and replicate them so that you have more of a specific DNA sequence to work with and do more genetic engineering and research with. And then gene editing. Gene editing enables precise modifications to the genetic code directly. One example is CRISPR. CRISPR stands for Clustered Regularly Interspaced Short Palindromic Repeats. Right, so CRISPR, this is a revolutionary gene editing technology that allows precise modification of sequences, and it has far-reaching implications for various fields, including medicine, agriculture, and biotechnology. I'll go over the figure in just a moment, but first I wanted to show you guys this video on how CRISPR works.
In a document, if we suspect we've misspelled a word, we can use the find function to highlight the error and correct it or delete it. Within our DNA, That function is taken on by a system called CRISPR-Cas9. CRISPR is short for clustered, regularly interspaced short palindromic repeats. CRISPR consists of two components, the Cas9 protein that can cut DNA and a guide RNA that can recognize the sequence of DNA to be edited. To use CRISPR-Cas9, scientists first identify the sequence of the human genome that's causing a health problem. Then, they create a specific guide RNA to recognize that particular stretch of A's, T's, G's, and C's in the DNA. The guide RNA is attached to the DNA-cutting enzyme Cas9, and then this complex is introduced to the target cells. It locates the target letter sequence and cuts the DNA. At that point, scientists can then edit the existing genome by either modifying, deleting, or inserting new sequences. It effectively makes CRISPR-Cas9 a cut-and-paste tool for DNA editing. In the future, scientists hope to use CRISPR-Cas9 to develop critical advances in patient care or even cure lifelong inherited diseases.
I did want to go over a little bit about how CRISPR works. Again, we'll go into lesson. But CRISPR is super cool. It's a gene editing tool that bacteria use, and scientists have figured out a way to use it too. It involves two main parts. There's a guide RNA called gRNA and an enzyme called Cas9. The gRNA is like a GPS that directs the enzyme, the Cas9, to a specific spot in the DNA, in the genetic code. And once the Cas9 gets to where it's supposed to go, it acts like a little pair of scissors, and it cuts the DNA at that spot. Then the cell's repair mechanisms will kick in, and it makes changes to the DNA sequence, like it will add or delete little bits of it, and it will turn off certain genes. And in this way, you have a very incredibly precise and powerful way to go into the DNA and edit genes. And you can totally change the game as far as genetic research and different genetic therapies.
So, what exactly is gene therapy? First, let's start with the basics. The majority of our body's genetic information is stored in 23 paired chromosomes inside the nucleus of our cells. Each chromosome is made up of DNA that stores information to determine our unique characteristics. Specific sections of DNA are called genes. As far as we know, humans have between 20,000 and 25,000 genes. Usually, a person has 2 copies of each gene, 1 copy passed along from each biological parent. Genes provide the instructions on how to make proteins. Proteins play an important role in how our body functions. And a small change to the DNA within our genes can alter how proteins work, which can then affect how a person breathes, walks, or digests food. To better understand how genes change, here's Dr. Hans-Peter Keim.
Gene variants, which are genetic changes, happen to everyone and occur as cells age or are exposed to certain chemical or environmental factors. These gene variants can also be passed along from parents. Cells often recognize these genetic changes and repair them. Other times, they can cause a disease or disorder, and treatment is needed.
That's where gene therapy aims to help, by addressing the underlying cause of disease. If genes are like the blueprint to our body, gene therapy can fill in missing parts or correct errors in the drawings. Gene therapy is the use of genetic material to treat or prevent disease. The genetic material used or targeted is DNA or RNA. DNA stores our genetic information, whereas RNA is a modified copy of that information needed for the cells to build the correct protein. In gene therapy, the genetic material is transferred to the cells and then changes how proteins are produced by the cell. It can reduce levels of certain disease-causing proteins, increase production of working proteins, or even produce new or modified proteins within the cell. The genetic material needs help to get where it needs to go, like being wrapped in a package with an address label. This package delivering the genetic material is known as a vector. Typically, viruses are used as vectors because they've evolved to be very good at getting into cells. But in this case, scientists use this same ability to deliver the new therapeutic genetic material into the cell. Any disease-causing part of a virus is removed, allowing it to enter the cell without making a person sick. All vectors are tested many times for safety prior to being used in humans. There are two main ways to give gene therapy: in vivo and ex vivo.
In vivo gene therapy means the gene is delivered directly into the person, such as through an injection. Ex vivo gene therapy removes the person's own cells and delivers the gene to these cells outside the body. These modified cells are then returned to the person. Deciding the right approach depends on the best way to target the disease.
There are still many challenges to overcome for those seeking gene therapy as potential treatment options. One challenge is time. It's a lengthy process to test potential treatments in clinical trials and then undergo the FDA approval process. But advances in gene therapy research allow us to better understand how we could treat and hopefully someday prevent rare and debilitating diseases.
There's other applications to the study of genetics. Genetics has numerous practical applications across various fields. Just in medicine, again, it's crucial for understanding the genetic basis of diseases. It enables the development of targeted therapies and personalized medicine. Some of the science on this is still pretty young, but in other areas it's very developed, so it's very exciting. Genetic testing is increasingly common. Genetic testing allows individuals to assess their risk for certain disorders and determine their ancestry. In agriculture, genetics is employed to improve crop yield. It enhances disease resistance and develop genetically modified organisms. Forensic genetics helps to identify individuals through DNA profiling, so this helps a lot in criminal investigations. And that's just a few of the uses. There are new ones cropping up every day. The study of genetics raises important ethical and social considerations, issues such as genetic privacy, discrimination based on genetic information, and the potential misuse of genetic technologies. And all of these things require careful consideration. It can be kind of tricky, and society really kind of needs to work to navigate these challenges while ensuring that the benefits of genetic research are accessible and used responsibly. That concludes my general overview and the introduction to my genetics course. It is a very interesting course because the study of genetics is fascinating, and it's transformed our understanding of life as we know it. It's paved the way for groundbreaking discoveries. It's unraveled the mysteries of heredity, provided insights into disease mechanisms, personalized medicine, and so many exciting things. Okay, well that's all for today. I really want to thank you guys for watching. Don't forget to do the reading, check the description for more videos related to these topics, and leave your questions for me in the comments below.